that consolidates two industry-proven technologies to perform both urine chemistry and sediment analysis in one small package. This total walk-away system minimizes labor-intensive manual steps and frees operator time for other valuable activities. The easy-to-use analyzer features one-step loading of trusted Ocean dry pad urine chemistry test strips with no calibration required. During analysis, the chemistry module aspirates the sample and precisely dispenses urine on each pad for measurement. The module measures 9 chemistries: glucose, protein, bilirubin, urobilinogen, pH, blood, ketones, nitrites, and leukocytes. For improved accuracy, the color compensation pad accounts for normal urine colors, and abnormal color detection notifies users of false positive reactions. It also performs specific gravity and color and turbidity measurements. The sample rack then advances, based on user-defined criteria, to the flow cytometry module. Here, a probe mixes the sample and aspirates into 2 separate chambers. The sediment chamber is heated to 35 degrees Celsius to remove any amorphous urates. The urine sample is mixed with diluent and fluorescent stain to accurately detect and enumerate formed elements in the sample. It can accurately detect and enumerate red blood cells, white blood cells, epithelial cells, and casts. The analyzer also flags 6 additional elements for review.
Is an integrated piece of equipment consisting of an incubator at 35 degrees to allow optimum bacterial growth, an agitator, the bottle racks rock continuously, and a growth detector system inbuilt into it. Blood culture bottles are loaded onto the analyzer and incubated for up to 5 days. When microorganisms are present in the blood culture, they metabolize the nutrients within the culture medium and release carbon dioxide into the medium. A dye sensor in the bottom of the vial reacts with the carbon dioxide. This reaction modulates the amount of light absorbed by a fluorescent material in the sensor. Each blood culture station in the analyzer contains a light-emitting diode photodetector, which measures the level of fluorescence every 10 minutes. A negative vial produces a linear graph as demonstrated. A positive vial produces an exponential curve graph as can be seen. Once the fluorescence reaches a specific threshold, the positive indicator light flags on the machine, and the biomedical scientist needs to remove the bottle to find out what is potentially causing the bloodstream infection. We cannot predict what might be present in a patient's blood culture, and therefore, in this setting, all manipulations of positive blood cultures need to take place in special containment equipment.
And is a technique for making millions of copies of a gene or a particular DNA target of interest. Copies are made using a very small initial quantity of DNA template, even down to just a single DNA molecule. PCR amplification means that detection and identification of your target DNA can be made using visual techniques based on the size and charge of the piece of DNA you have amplified. The theory behind every PCR, whatever your DNA target, is the same. A mixture is created that contains 5 basic things: your DNA template, a DNA polymerase enzyme, forward and reverse primers and nucleotides, also known as dNTPs, and buffer to stabilize the enzyme. For each new PCR, you choose the primers and design them to be complementary to short sections of DNA next to or near your region of interest. Usually they are between 20 to 30 bases or nucleotides long. The enzyme is there to extend the primers, which have annealed to a specific region of your template, and use the free nucleotides in the mixture to build the new strand complementary to your target. The primers should only anneal to this particular region of interest. During the PCR, the DNA being amplified is heated and the double strands separate. Upon cooling, the primers bind to the template and the process of adding complementary nucleotides by the polymerase can begin. The initial heating step is called denaturation and usually occurs at 94 or 95 degrees centigrade. It needs to be at this temperature long enough to denature the template completely. The following step, when the primers are able to bind to the template at a lower temperature, is called annealing. This temperature depends on the TM or melting temperature of your specific primers and is usually between 50 to 65 degrees centigrade. This step doesn't need to last very long, typically 10 to 30 seconds. The final step in this first amplification cycle is the extension at 72 degrees centigrade. This is the optimum temperature for the DNA polymerase to build the complementary strand, following on from the 3' end of the primer, using the free nucleotides. The polymerase adds them at around 50 to 100 bases per second, so the duration of this step will depend on the length of your target, but 30 seconds to 1 minute is usually long enough for most products. This process of denaturing, annealing and extension is repeated 30 to 40 times, theoretically doubling the number of DNA copies at each cycle, and thereby increasing exponentially the number of copies of your target of interest in the mixture. Samples can be prepared and incubated in a thermal cycler, and a complete PCR reaction can be performed in a few hours, or even less than an hour with certain types of rapid instrument. The lab area used to set up and run your reactions needs to be carefully controlled and maintained. PCR can detect a single molecule of DNA, so it is essential to have a unidirectional workflow with, ideally, 3 separate rooms in your system. Firstly, you must have a clean room. This room would contain all of the reagents for your PCR except the DNA template. None of the reagents and consumables would have been into the area where PCR products are generated or analysed. The best system would involve a workflow where the clean room would not be revisited on a day when you've been in other potentially PCR product contaminated areas. The second room in your system would be the extraction lab where you've prepared your DNA template ready to put into the PCR. Once you've set up your reaction in your clean room, you would take the reaction tubes or plates to the extraction lab and add your DNA template. From there you can go on to the third room, the PCR machine room. This is where the PCRs take place and where the products are usually analysed by visualisation on a gel or by other methods. It is best to consider that PCR products that you generate contaminate this third laboratory, however careful you are, so never set up a PCR reaction here or take reagents or consumables from this lab into the other rooms, or they could contaminate your clean system.
One-step approach and the two-step approach. In two-step RT-PCR, the first step involves the reverse transcription, or RT, of RNA by a reverse transcriptase. This reaction generates the first strand complementary DNA, or cDNA. cDNA is then transferred to a separate reaction tube for the second step of this process. During the second step, the second strand cDNA is generated and amplified by a DNA polymerase. So, in two-step RT-PCR, reverse transcription and PCR occur in separate reaction tubes. This approach is used when there's a need to analyze multiple targets from the same RNA sample in separate reactions or to store synthesized cDNA for later use. In the other approach, known as one-step RT-PCR, the reverse transcriptase and DNA polymerase are pre-mixed, which allows the RT and PCR steps to be performed in the same reaction tube. Not only is the one-step RT-PCR approach faster, it also requires less pipetting and minimizes possible contamination, thereby improving data reproducibility. It also enables easier processing of large numbers of samples for high-throughput applications.
To begin the reaction, the temperature is raised to 95 degrees. At this temperature, the double-stranded DNA is separated into single strands. Then, three major steps are repeated during each cycle. First, denaturation to obtain single DNA strands. Second, annealing. The temperature is then lowered to approximately 60 degrees. This allows the primers and the fluorescent probe to bind to your target sequence of interest. And third, extension. Between 60 and 72 degrees, the polymerase extends the primers and cleaves the probe, which generates an increase in the fluorescent signal proportional to the amount of amplified product. After one cycle of PCR, there are twice copies of the target DNA. These steps are repeated through around 45 cycles. When the starting material is RNA, one first additional cycle of reverse transcription is required. The real-time PCR reaction requires the following components: Taq DNA polymerase, reverse transcriptase, primers, probe, DN-TPS, MgCl2, magnesium chloride, buffer, stabilizers, internal control. ViaSure real-time PCR detection kit contains the following components: well strips with all the previous reactives in a stabilized format, positive and negative control, rehydration buffer to reconstitute the stabilized product, water RNA SE DNA SE free, optical caps for sealing wells during thermal cycling. Molecular diagnosis unit consists of 4 areas: nucleic acids extraction, pre-PCR, setup, PCR, amplification and results interpretation. The first area aims to process potentially infectious samples and subsequent extraction and purification of nucleic acids with the RNA-DNA ViaSure extraction kit. We recommend to dilute stool samples before extraction and use the supernatant to perform DNA-RNA extraction. Homogenize the fecal sample, collect a pea-size and transfer to a 2 milliliter vial with 200 microliter of RNase DNase free water, PBS or physiological serum. Vortex the sample and centrifuge. Samples are lysed at elevated temperature in the presence of lysis buffer and proteinase K. Transfer the supernatant to the 2.0 milliliter collection tube. Then add lysis buffer and proteinase K and vortex vigorously. Incubate 10 minutes at 65 degrees and then 10 minutes at 95 degrees. After optimal binding conditions are adjusted by the addition of isopropanol, the lysate will be applied onto the mini-spin column and DNA-RNA bonds to the surface of the filter membrane. Add binding buffer and mix by pipetting or vortexing. Incubate the sample at room temperature and transfer the lysate onto the column mini-spin column set. Centrifuge and discard both the filtrate and the RTA collection tube. Then place the column mini-spin column into a new RTA collection tube. Contaminations and enzyme inhibitors are efficiently removed during the following wash steps, while the nucleic acids remain bound to the membrane of the mini-spin column. Add wash buffer 1, centrifuge and discard. Add wash buffer 2 and centrifuge. Repeat this washing step once. Centrifuge to remove ethanol residues and discard the filtrate and the RTA collection tube. The nucleic acids are finally eluted from the membrane using elution buffer. Place the column mini-spin column in the sterile 1.5 ml collection tube. Add elution buffer preheated at 65 degrees. Incubate at room temperature and centrifuge. Discard the column mini-spin column. Store the DNA/RNA sample at 4 degrees for immediate use or preserve from -20 to -80 degrees for longer periods. The sample is ready for the next area. Take the ViaSure Pathogen Real-Time PCR Detection Kit and separate the required number of wells, including samples and 2 reserved wells, for the positive and negative controls that must always be included in each assay. If necessary, cut the required number of wells with the scissors and remove the protective aluminium seal. Reconstitute each one of the wells with 15 microliters of the blue vial rehydration buffer. Finally, add 5 microliters of the purple vial negative control in the reserved well for negative control. After this process, move the rehydrated well to the next area. 2 independent processes are made in the third area. First, the positive control rehydration. ViaSure Pathogen Real-Time PCR Detection Kit contains a positive control that must be rehydrated before using this assay. It is recommended to open and manipulate before starting with the assay. Reconstitute the red vial positive control with 100 microliters of the white vial RNase, DNase free water and mix with the help of the vortex. Store at -20 degrees after resuspension and divide in aliquots to minimize the freeze-thaw cycles. The second process is the addition of the nucleic acids and positive control to the rehydrated wells. Add 5 microliters of extracted DNA, RNA from each sample in the rehydrated wells, with the exception of the 2 well reserved for the positive and negative control. Add 5 microliters of reconstituted red vial positive control in the reserved well for positive control. To finish, divide the required optical caps number and close the wells. After this process, move the plate to the real-time PCR equipment. Place the plate in the thermocycler, program the thermocycler and start protocol. When finished, analyze the results with the software installed in the equipment. ViaSure provides maximum flexibility and compatibility with leading open system thermal cyclers and the latest technology for the stabilization of products.
a new type of coronavirus has emerged from the city of Wuhan in China. Within weeks, it has spread to several countries and has killed multiple people. Corona refers to the virus's distinctive shape. They are a large group common among mammals and birds. But in rare cases like this one, they can spread to humans, a process known as zoonosis. This new virus is the 7th known human coronavirus, and it affects the respiratory system. Some coronaviruses have mild symptoms, however others are much more dangerous. SARS and MERS are both coronaviruses.
When designing a LAMP experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis, initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential LAMP amplification. This LAMP dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatemers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.